the discovery that I hope will lead to something for Lyme is that every cancer cell turns on its surface a molecule that is a don't eat me molecule for the kinds of cells in the body, the scavenger cells that are supposed to scavenge bacteria, viruses, cancer cells, infected cells, dying cells. But the cancer cells put on a don't eat me signal, so they can't eat. We began looking at some viral infections, and we found that virtually every virus-infected cell, if it lasts for a long time in the body, protects itself from the eating cells by putting on CD47. Some viruses, in their coat protein, even though they're little tiny active things, make a coat protein that looks to the macrophage like the don't eat me signal. And so they stop themselves from being eaten by macrophages. We've developed an antibody which is now in clinical trials for cancer that blocks the don't eat me signal. And so our part of looking at this disease is that the disease has beaten the immune system so far. And how is it beating the immune system? Well, if it is doing it in part by preventing the macrophages, the big eating cells, from eating either the bacterium by itself or, more interestingly, those spirochetes that live inside cells. If it gets inside a cell and hides from the immune system, then no matter how many T cells and B cells you have making an immune response, it's the infected cell in hiding that would keep reintroducing the Lyme disease agent to the body. So we're testing that. We're testing with reagents that would recognize a mimic of the don't eat me signal and other reagents that would cause infected cells that avoid being eaten to be eaten. This is a little bit of a long shot. We're trying to find out with the best reagents we have. If we see any hint of effectiveness, then we will look for the few patients who have a cancer like a lymphoma and also still have Lyme disease, so that they could be treated for their lymphoma with a therapeutic dose of our antibody, and we could measure, did we, in a real patient, get rid of the Lyme agent. That's what we're trying to do. We know it's a long shot, but we think it has a chance. We'll be first to say, don't give us any more money if it looks like it's not working. So they didn't look at it because everybody does what they're an expert at. So if you're a T cell biologist, you can look at T cells. If you're an antibody person, you can look at antibodies. If you're an antibiotic person, you're going to look at that. So this is an opportunity for people in many different disciplines to join up to talk about and then look for potential therapeutics or new ways to treat the disease. This happens all the time. I mean, you would have thought that people would have thought that if macrophages are eating cells and they kill bugs and other stuff, maybe the bugs had a defense against it by providing don't eat me signals. Metchnikoff discovered macrophages in the late 19th century, and nobody came up with this hypothesis until we saw that human leukemias had put this don't eat me signal on its surface, and now we know every cancer does. So we are looking at cancer. That's our primary objective, and we're taking a new look at persistently infected cells, whether they're infected with Lyme disease or HIV or malaria or tuberculosis. We're looking at that whole set of things to see if we had missed this perfectly simple principle that if you block the don't eat me signals, what happens? Will they get eaten and killed? In the 1980s, in studying the blood-forming cells in mice, we found that we could isolate blood-forming stem cells in mice. And we proved that they would reconstitute by transplantation the whole blood-forming system in another mouse that had been irradiated or was in some very difficult situation. So we transplanted blood-forming stem cells in mice. And then we formed a company and did it in humans. And then we found at that time, in the mid-90s, that we could treat women with metastatic breast cancer by isolating in pure form their blood-forming stem cells away from any cancer cells that might be in their tissues, then treat them with an otherwise lethal dose of combination chemotherapy and rescue them with cancer-free stem cells. Now, there wasn't a single oncologist or bone marrow transplanter in the world who believed this would do anything. And now, 18 years later, a third of the women we treated are alive. We've done this with isolating brain-forming stem cells, and now are treating in clinical trials people with spinal cord injury and people with macular degeneration. And we treated a couple very rare diseases of childhood, and that looks very promising. So I believe that it is the job of scientists to think about how their discoveries could be translated. I grew up in an era of science where we thought that's the last thing we should think about because it might divert our thoughts away from the basic science we're doing. That's wrong. What's the right thing to do is to get everybody to try to think how a discovery they made, even though they think it's unrelated to clinical medicine, to think about how it might be translated. My institute is built on that premise. For the blood-forming stem cells alone, we've hired in several people to be expert in transplanting pure blood-forming stem cells in children to cure genetic diseases of the blood like sickle cell anemia or Mediterranean anemia or severe combined immunodeficiency, and also diabetes and lupus and a whole bunch of other diseases of blood-forming cells. So we not only have made the discoveries, but thanks to being at Stanford, we've been able to develop clinical trials here at Stanford to find out if our idea is right. We just, with the help of the Children's Hospital and the Adult Hospital and the Stanford Medical School, built a cell sorting facility so that we can get pure blood-forming stem cells for clinical trials. We're the only place in the world doing this. We've hired people who are cancer experts who find cancer stem cells, and our antibody to the don't eat me signal is just one of the many therapies that are going from the research laboratory to preclinical test of principle to clinical trials. And as I mentioned before, we will be looking, if we have any good hint that we're getting rid of the Borrelia, the Lyme agent, with anti-47 in animal models, we will be looking for patients who have a malignancy we can treat with our antibody and also have Lyme disease. That's the way that we can, without big risk or huge expense, get a hint of whether we're going down the right track, not just in animal experimentation, but in real humans with the disease. Most medical schools don't try to have large research units that do discovery on the basics of disease causation. And even fewer have research groups that believe it is their job to translate. And even fewer set up the clinical trials group so they can translate. So I think Stanford is one of those places. Not a lot more. There are other great places, of course. I'm not saying that Stanford is the greatest. But if you think about it, this is a major medical school with major hospitals, surrounded by a major university which has computer scientists and physicists and engineers and chemists and biologists and pathologists and immunologists and stem cell biologists, all within a few hundred yards of each other. Eating lunch, their students are meeting with each other, so this is a unique place in that regard. When I talked about the ivory towers of people doing research for research's sake, and maybe thinking it's even a sin to try to think about how it would help a disease, Dean PIZZO realized that came from departments, and departments that reiterate training and hiring of people in their own fundamental discipline. In a way, departments are a way that people talk only to themselves and not to others. He broke that up by making institutes that cross departments. And that was the fundamental way that he could start to change the culture at Stanford. If you make a definition of the private sector as saying the real important part of the private sector is those people who are in disease advocacy groups. We never would have gotten Prop 71 across without the strong support of disease advocacy groups. And when Bob Klein and I and others wrote Proposition 71, we made sure that at every level of governance and review, people from disease advocacy groups were there to remind the reviewers and the scientists why we're doing this. It's critical. It's easy to go off track. President Bush was thinking about his own religion and his views of morality, and he made an executive order that in effect stopped stem cell research in many areas that we now know are incredibly important. It was a grassroots movement begun by parents of diabetics who said, just a second, will that stopping that research stop something that could be good for my kid someday? And we said, yeah, because actually we know about stem cells and blood-forming stem cells and the need to find insulin-producing islet-forming stem cells. And sure, the likely long-term source of both of those kinds of cells so that you can replace a diabetes-prone blood-forming and immune system with one that works, and also replace the islets that were lost, and not require expensive and life-threatening drugs. But we said to the public, because I gave a lot of the talks, that no therapy comes less than 10 years from the discovery. There's a long process of discovery, preclinical testing, then large funding to move toward and do the early clinical trials. And the late clinical trials can take a long time, and then the therapy is out there. But if you don't start now, then you mean that you're putting it off for another 10 years and another 10 years. This was a states' rights movement because the federal government took away something that the state thought was important. So we, as people in the state, developed it. And this is the first time that a government agency is taking on the full costs of preparing for and doing early phase clinical trials. Before, we would have to, just as I had to, you'd have to go form a company. And then you would have to have a CEO whose main job was raising more money for the next step and the next step of the company. And as more money comes in, it always demands a seat on the board. And after a while, the board is made up only of those people who had the money. And then eventually they make decisions as if they knew the science when they're going after the money. So usually, money and the ability to turn around the science to make more money is the driving force. Whereas we in medical schools believe that you try to advance knowledge to improve health. That's a problem we haven't really looked at in the face. And when you have a federal government that can be so influenced by private interests and their own religious or political interests, to stop promising avenues of therapy, not just for themselves or their family, but everybody, then you know you're in a very bad situation. You got to bring it all together to try to change this incredibly difficult and slow and very expensive way to develop very high-cost medicines for the few. So we got to move it in order to find and involve the people who make the discoveries, because they're the ones who can find the glitches that come up on the way. So Prop 71 and the California Institute of Regenerative Medicine is the first group ever to do that. They're focused on stem cells. We're getting a lot of cancer treatments out of that. We hope that we'll get infectious disease treatments also. But I think this is a good example. The very key to making discoveries is to be training people who can make discoveries. And the key here at Stanford is to try to make sure that we interest the very best and the brightest who are pursuing both scientific and medical training careers to keep in it instead of going out to where they can make a lot of money. Well, we have pending here at Stanford an initiative to greatly broaden the research experience for people who want to come to Stanford, and telling them you won't get out in 4 years. You can't just memorize your way through and then go do a residency. If you come to Stanford, or if you opt into one of these plans at Stanford, it may take you 5 and sometimes 6 years. You won't get a PhD, but you'll know enough which area of science needs to approach the problem that the patients you've seen bring to you. So we're at a time when people trained in both medicine and science, the federal government is cutting support. NIH, when it tries to make its budget, found that an easy place to cut. So there's nobody looking after this. I came to Stanford Medical School. I know I'm not young, but I only applied to Stanford Medical School because it was the only school that had 5 years for medical school. And it took the first 2 years, which are usually 8 hours a day of classes and 8 hours a day of study and a couple hours to sleep, and said, we'll spread out that basic science. We'll make sure that everybody has a half a day every day for 3 years to pursue their research interest. Now, many years later, I went back and looked at my class. Half of them had full-time careers doing research in medicine at the various medical schools around the U.S. Harvard's never got better than 20%. So, knowing where you have to get the right minds in to figure out how you join medicine and science, and look at every patient you see from the point of view, what is the unanswered need for those patients, whether it's an economic need or a social welfare need or a scientific need. So yes, if you really get hundreds of millions of dollars, save some of that money to attract really young and bright minds to come to the only school that'll have that program. Stability of funding for 5 years is very important. The way that Prop 71 and CIRM was established was a bond issue, so you guaranteed 10-year stability of the agency to fund. And you can't go back to the legislature every year to ask for it because you got the bonds already. So, yes, finding one or another way to get stability of funding so people can go in and realize it's worth taking the risk to go out of their comfort zone and do Lyme. But the second one I would say, just to add to it, is I would encourage those labs you fund at Stanford to recruit one medical student each who will spend the extra time learning about the medicine of the disease and the science of the disease. One of the things that I learned when I first was on the Scientific Advisory Board of Amgen and DNAX was that was the first time in my whole career that I sat in the room with biochemists and docs and protein biochemists. And so when you break down those barriers, as PIZZO did to form the institutes, and as you are doing to bring people together who don't talk to each other usually, is to get that cross-disciplinary discussion and to bring their trainees into understanding that you're going to go a lot further with that cross-disciplinary discussion. And of course, by, again, the stability of funding so that they don't have to run out and spend, as we do for NIH as our source of money, a very significant part of our time rewriting and rewriting and rewriting grants. That is the worst waste of our time. So yeah, if you can do that, that's terrific. Oddly enough, the review of grants at NIH, you won't believe it when I say it, is mainly aimed at asking the question, will the idea work? Rather than what's the track record, the recent track record of the person who's trying to make a discovery and work in a particular disease. If you go to IBM or Bell Labs or any of the places, you would say, well, would you hire people on the basis of what they promised you that they could do? The experiments they could do? The answer is no, because anybody who's good in science knows within a year that you start doing something that you said you were going to do, you learn something from the experiment or from somebody else's experiment that makes you change your hypothesis. And you've got to look for the kind of person who can adapt to the data and change the hypothesis. So NIH is mainly funding things in the wrong way. And because they're not paying attention to people who have been successful at making and translating discoveries, there's a lot of waste of money. So you can imagine now we're getting to a perfect storm. When in 2008, a lot of money left the country, and all federal agencies had to worry about saving the big banks, and money for NIH was going down. We also had these study sections that were using that limited amount of money. It used to be 25% of approved grants, 18, 15, now it's 7%. And the people they're funding are not the ones who are advancing discoveries all over the country. I know of great labs, Nobel laureate labs, leader of the field labs that are shutting down because federal support is irrelevant to them. They get rejected when they put in their grants. look at some of the great advances that have happened with CERN. Where, as I said, disease advocates were there to make sure things were funded. Where discoveries and the people who made the discoveries moved all the way to clinical trials without having to go get venture capital and form a company. And you, at the university, build up disease teams where now everybody on the team learns what it takes to do a clinical trial. We're educating that whole group. So I would say that the way to convince people that there's a different way to fund science is to point at those examples where things go forward fast. Our antibody to CD47, our anti-cancer trial, all of that came from a discovery, and essentially our first paper was 2009. By 2014, we were in clinical trials. We did it ourselves, we built up the team ourselves, and the team knew how to do it. We had a lot of glitches, and because we knew the biology of the system, we knew which ones were serious and which ones we could get around. The current U.S. way of translating discoveries, forming biotechs or letting big pharma do it, well, biotechs are always raising money, so they spend a lot of their resources raising more money. Big pharma are making money. They found the most efficient way for small molecule drugs, so they create silos, discovery, preclinical proof of principle, operations, clinical trials early, clinical trials late, and they don't talk to each other because they're too big to talk to each other. And at the very back end, they have directors, who say, as they should say, well, our main obligation is to make a profit for our shareholders. They're not sitting in that room saying our main obligation is to advance medical science for the health of people. So those are the lessons that we've learned in this last 10 to 15 years. Those are the lessons that I hope we can pass on rather than going back to the old way, which makes medicine expensive and really not advancing at the rate it should.